The overall goal of this procedure is to evaluate the effects of genetic and pharmacological manipulation on preference for dietary fat, which is a key factor underlying overconsumption of food and obesity. This is accomplished by first filling hoppers A and B with 10% fat food and measuring food consumption 24 hours later. The second step is to switch the positions of hoppers A and B and weigh them again the next day to determine the baseline feeding position bias. Next, the fat preference can be determined by repeating the above steps multiple times with 12.5% and higher fat food. The final step is to calculate the preference score by dividing the grams of intake of the higher fat diet by the total food consumed of both diets. Ultimately, the fat preference model is used to show how experimental manipulations may change a rat's preference for high-fat food.
This method can help answer key questions in the obesity field, such as what pathways in the brain mediate high-fat feeding behavior.
This technique can be extended toward the development of therapies to treat obesity and eating disorders, in particular because we can selectively reduce intake of foods that are high in fat and sugar.
For the following protocol, male Sprague-Dawley rats are singly housed and kept on a 12-hour light-dark cycle. Fill the food hopper with a low-fat food and habituate the rats to the food and colony room for at least 7 days prior to experimentation. For each of the 3 days before taking baseline measurements, handle each test animal to reduce stress.